Hello, we are students from Ratchasima Witthayalai School.
Here to present our project title.
Persistence of touch DNA, exposure to different environments. We have divided the presentation into 5 sections. In crime scene investigations, forensic evidence plays a crucial role in solving cases. One notable example is a case from July 15, 2015, where an assault with a deadly weapon resulted in a tragic loss of life. In this case, a glass became key evidence. Investigators discovered that touch DNA, a trace amount of genetic material left behind when a person comes into contact with the surface, was present on the glasses. Though invisible to the naked eye, touch DNA can provide critical clues. However, its minute quantity and susceptibility to environmental factors make detection and analysis challenging. Currently, forensic science has developed advanced methods for detecting touch DNA, mainly corneocytes, by staining sample with a fluorescent dye that is specific to nucleic acid, named diamond nucleic acid dye or DD in short. These samples are then visualized under a portable fluorescent microscope, which can detect the stain cells on various forensics items, such as glass, bullet casings, and firearms. As a result, latent touch DNA become visible as green dots as shown in the image below. One challenge in using DNA as forensic evidence is a tendency to degrade over time. Environmental such as temperature, humidity, ultraviolet radiation, and exposure to rain can significantly affect the preservation of DNA. This condition causes DNA to degrade at varying rates, making it difficult to obtain reliable results. Therefore, this project focuses on studying the effects of these environmental conditions on the persistence of touch DNA. The objective of the experiment are 1. To evaluate the impact of environmental factors on the quantity of DNA of various durations. 2. To improve the efficiency and reliability of DNA detection methods while reducing the expenses associated with analyzing samples with insufficient DNA quantities.
In the experimental methodology, the first step involved DNA deposition. The sum values put on the gas slide for duration of 15 seconds to ensure sufficient DNA transfer onto the surface. The second step was to place the experimental setup into the 6 environmental, consisting of 1 indoor with low temperature, 2 indoor with dark ambient, 3 indoor with ambient light, 4 indoor with high humidity, 5 outdoor with clear cover, and 6 outdoor. The experimental results were collected every 0 to 4 and 8 weeks in each environmental. The next step is to dye the gas slide with diamond nucleic acid dye. 10 microliters of diamond nucleic acid dye was prepared onto the gas surface to stain the sample. The gas surface were examined using a dyno-light fluorescent microscope at magnification of 50 times and 220 times. DNA persistence was assessed counting visible cells manually within 3 frames per millimeter square at magnification of 220 times of its gas slide.
When the fingerprints under various environment and time were tested for persistent DNA using fluorescent staining and viewed under a portable fluorescence microscope, it was found that all the samples exhibit different visualized fluorescent spots, especially between low temperatures and outdoor conditions. When the persistence of DNA was assessed by counting the number of visualized cells, it was found that at the initial time point, our environment started with 28 cells per millimeter square in average. When the time passing through week 8, DNA persistence varies significantly among the various conditions, as you can see in this graph, which show the number of stem cells over time from week 0 to week 8 in 6 conditions. At week 8, low temperature conditions show the lowest lot of cells, about 27 cells per millimeter square, or only 4% degradation. At dark ambient conditions, 17 cells left, or 39% degradation. At ambient light conditions, 12 cells left, or 57% degradation. At high humidity conditions, resulted in a sharper decline of 7 cells left, or 75% degradation. Under regular cover conditions, 5 cells left, or 82% degradation. While other conditions, only 2 cells can be detected, or 93% degradation. Next, we will discuss how temperature has an effect on preservation of DNA. The results show that the number of cells in low temperature is higher than dark ambient due to low temperature can slow down cellular processes, including metabolism and degradation, which could preserve the stained corneocytes over time. Next, Humidity negatively impacts DNA preservation. In high humidity conditions contain more water molecules in the air, which increase the likelihood of hydrolysis bond forming. This hydrolysis bond cause nucleotides to detach, leading to the degradation of DNA strand. Next, ambient light also negatively impact DNA preservation. This is due to light from LED bulb, which typically have the frequency that can trigger the formation of covalent bond between adjacent pyrimidine base. This process generates two types of light-induced products, cyclobutene pyrimidine dimers and pyrimidine pyrimidine photoproduct, which ultimately cause damage to the DNA strand. The other also negatively impact DNA preservation, with the reason similar to the light ambient. While both are influenced by ultraviolet light, outdoor environment introduce additional factors such as temperature fluctuations and exposure to humidity. As shown in the graph, DNA degrades faster outdoor, highlighting the compound effect of sunlight and humidity. DNA level were lower in the open environments compared to the covered one and was lowest among all conditions, emphasizing how exposure to the direct environment elements such as precipitations and therefore accelerates DNA degradations, further highlight the challenge in preservation of DNA evidence in outdoor crime scenes.
In conclusion, DNA persists best in low-temperature indoor environmental with only 4% degradation, while outdoor environmental cause lipid breakdown with 93% degradation. This finding helping forensic practitioners prioritize its evidence, enhancing DNA profiling and improving the process of DNA detection from fingerprint in forensic science, particularly in the initial screening of biological evidence beyond to detailed DNA analysis. There are our acknowledgement. There are all the reference. Thank you for your attention.